Hello everyone, welcome to Cookie School. So today we are going to discuss about the chapter Cell Cycle and Cell Division. So here totally there are 5 topics in this chapter. Okay. So how a cell is dividing we are going to see, and what is the cycle of cell we are going to see in this chapter. The first one is cell cycle. Okay. In that division is there where phases of cell cycle. Okay. And the second topic is M phase. So here the M phase is known as a mitotic phase. So here there are 4 stages mainly, and the last one cytokinesis. Okay. Prophase, Metaphase, Anaphase, Telophase, and cytokinesis. Okay. And the third one is significance of mitosis. So what is the significance of mitosis? And the fourth one is meiosis. And in meiosis there are 2 subtopics, meiosis 1 and meiosis 2. Okay. And fifth one is significance of meiosis. Okay. So first one is cell cycle, mitosis, significance of mitosis, meiosis, significance of meiosis. Okay. So we'll discuss about cell cycle and phase of cycle. So now we are going to see the first topic, which is cell cycle. Okay. So here cell cycle is nothing but the various stages in cell. So what are the cycles the cell is going on, in which stage what cell division happens, and in which stage the cell grows. Everything we are going to study here. So here during cell division, both DNA replication and cell growth take place. Okay, during cell division. See here, cell cycle is generally what we are going to see in cell cycle. Cell duplicates its genome. Genome is nothing but its genetic material, which is the chromosome, which is RNA or it will be DNA. So cell duplicates is nothing but it replicates. It replicates means it breaks them into two. Okay. So it replicates its genome. Synthesis of other constituents of cell. So other constituents of cell are nothing but the mitochondria, Golgi complex, endoplasm. So various other organelles in a cell is known as constituents of cell. Okay. So it also synthesizes the other constituents of the cell, and the division of 2 daughter cells. It divides into 2 daughter cells. Okay. So here these all takes place in cell cycle. Here we are going to see about these things. So cell growth, if you take cell growth generally the cytoplasmic growth. Okay, it is a continuous process where throughout the life the cell grows. Okay, so where DNA synthesis occurs only in one particular stage. Okay, so DNA synthesis is nothing but replication of DNA. To replicate, it has to synthesize and replicates. So that occurs in only one stage of life. Okay, but cell growth is a continuous process. It has to grow. So now what are the phases of cell cycle? If you take human cell, it divides 1 hour in 24 hours. Okay. But in yeast, the cell cycle is completed within 90 minutes. Okay. So this cell cycle is divided into 2 phases, interphase and M phase. Interphase is nothing but between 2 M phases. M phase is nothing but where cell division takes place, mitosis, equivalent division. Okay. So equivalent division is known as mitosis, where it divides into the cell, divides into is known as M phase. So between 2 interphase nothing but between 2 M phases. Between 2 M phases nothing but one phase it has divided. So and after next division it takes place, like this cell will divide to next 2 cells. So before that, what are the changes are happening is known as interphase. Okay, between 2 M phases is known as interphase. So this interphase lasts about 95% of the cell cycle. 95% of the cell cycle is interphase only. Whereas 5% of the cell cycle is M phase. Okay. So M phase is nothing but division, which takes, for example, if you take human cell, only 1 hour takes place for mitosis, whereas 24 hours, the rest 23 hours are interphase. Okay. So this mitosis have 2 kinds of divisions. Okay. One is nuclear division and another one is cytoplasmic division. Nuclear division takes place first because the nuclei has to separate, and then cytoplasmic division is nothing but the division of a cell into 2. The cell structures into 2. It is known as cytoplasmic. Into equal parts. Yeah, the other cytoplasmic components will divide into 2. That is known as cytoplasmic division. Okay. So this nuclear division is also known as karyokinesis. Okay. Karyo is nothing but nucleus and kinesis is nothing but division. And cytoplasmic division is nothing but cytokinesis, where cyto is the cytoplasmic. We learn about cytokinesis in this lesson. Yeah, we will be learning. And the cyto is nothing but the division of cytoplasm, and kinesis is nothing. Kinesis is division and cyto is cytoplasm. Okay. So karyokinesis is division of nucleus and cytokinesis is division of cytoplasm. So here we are going to see in detail about these phases. Okay. What are the interphase and M-phase we are going to see. So now we are going to see the phases of cell cycle, which we have already seen. So this is a cell cycle, phase of cell cycle. So imagine this is the cycle where a cell undergoes. Okay. So here if it starts from the M phase where the cell division occurs, mitosis phase, if a cell first starts to divide, it undergoes prophase, this one is prophase, metaphase, anaphase, telophase, cytokinesis, and then G0. Here the division completes, it ends at cytokinesis after the division of cytoplasm. So it becomes two daughter cells. When a small daughter cell undergoes growth and becomes, it requires the characteristics to divide again. Okay, so it grows. So this stage is known as G0, which is known as, so here G0 will be discussing later. And then here after G0 comes G1 phase, and then S phase. G1 is nothing but gap 1 phase. Okay. And then S is nothing but synthesis phase. G2 is gap 2 phase. So from here to here the cell does not divide. It undergoes growth and synthesis of all the components.
Ready to go.
Yes. Okay. So here now we are going to see about the interphase. So these prophase, metaphase, anaphase, telophase, cytophase all comes under M phase, which is mitotic phase, which is division. Here we are going to discuss about interphase. Okay, interphase is nothing but between two divisions what a cell undergoes, we are going to discuss here. So interphase is divided into three: gap 1 phase, synthesis phase, and gap 2 phase. These three. Okay. So here the cell is preparing for division by its growth and DNA replication. Okay, so cell growth and DNA replication. So here first we will see about the G1 phase, which is gap 1 phase. So here this phase, gap 1 phase, is between mitosis and synthesis phase. It is between mitosis and synthesis phase. This gap 1 exists. Okay. So this gap 1 is metabolically active. It continuously grows in this gap 1 phase. When the cell undergoes this gap 1, it continuously grows because it's small, and it is metabolically active. It is very active, but it do not replicate its DNA. Okay, it just grows with its own DNA, which it got from its mother cells. Okay. So after G1 gap 1 phase comes the S phase, which is known as synthesis phase. Okay. So in this synthesis phase only DNA replicates. Okay. So DNA synthesis starts. So the amount of DNA per cell doubles. So at the starting stage, and if you compare the starting stage and the stage, the initial amount of DNA and the final amount of DNA here. So here the amount will be doubled to this one. With respect to
this will be doubled by that one.
Synthesis phase replicates its DNA and doubles the DNA. Okay. So initially, if the amount of DNA is, if you, for an example, if you keep it as 2C, C is a constant. Okay. Initially, it has 2C DNA, 2C amount of DNA, and finally it has 4C amount of DNA because it doubles into 2. It doubles. Okay. So here no increase in chromosome number. Okay, so chromosome number, if it has 2n in this phase, here also 2n will be there. If it has n chromosomes here also n chromosomes. But the amount of DNA alone is doubled in the S phase. Okay, and this in animal cells, centriole duplicates in cytoplasm in the S phase. Okay, centrioles duplicate, they replicate themselves in this S phase in cytoplasm. So and then after the synthesis phase comes the G2 phase, which is gap 2 phase. So here proteins are synthesized in this phase. Proteins are synthesized which are required for the cell division. Okay, proteins are synthesized and cell growth continues here. Okay, so can you remember all these three phases? The first one is G1 phase, where the cell continuously grows and it is metabolically active but do not replicate its DNA. In synthesis phase, it replicates DNA, the amount of DNA is doubled, but it does not increase in chromosome number.
Okay.
And in G2 phase, the proteins are synthesized and cell growth continues. Okay. So we discuss some more characteristics of phases of cell cycle. What are the other unique characteristics of cell cycle? Yes.
Unique features.
So now we are going to discuss about some more unique stages of cell cycle. So here, what are the unique stages? So some cells do not divide. We have seen some cells which do not divide, and it replaces only damaged cells. For example, in heart cells of human beings, they only replace the damaged cells. They do not replicate. So some cells in adult animals do not undergo division. Example, heart cells. So some cells divide occasionally to replace the lost cells in the injury or death. So sometimes only they divide. So what these cells do? They exit their G1 phase. G1 is the gap 1 phase, growth phase. They exit that one and they come out of that phase and enter into an inactive stage called quiescent stage. Okay, it is an inactive stage called quiescent stage, which is G0. It is also known as G0. So here cells are metabolically active but do not divide unless called to do so. When it is needed, when it needs cells, it will divide. But until it does not divide. Okay, so this is the quiescent stage. Okay. So next in animals, mitotic division is seen only in diploid cells. Okay, diploid is nothing but 2n number of chromosomes, whereas haploid is n number of chromosomes. Okay. So in plants, this mitotic cell division is seen both in haploid as well as diploid. Okay. So what is mitotic cell division? Mitotic division is nothing but it retains it is equivalent division. If 2n number of chromosomes are present in the cell, after mitotic division, in each cell, in the two daughter nuclei also 2n number of chromosomes, 2n number of chromosomes will be present. Whereas in case of meiosis, it is not. Okay. So this is the mitotic cell division where it separates into 2n, each 2n number of chromosomes. Okay. So this one is equivalent division. So mitotic cell division. So next we are going to see the second topic, which is M phase, which is mitosis. And there we will be seeing many phases, how with mitosis the cell divides. Okay. So now we are going to discuss about our second topic, which is M phase, which is also known as mitotic phase, where mitotic division takes place, which is equivalent division. So here this is also known as equational division. So why known as equational division is because number of chromosomes are present. The number of chromosomes are same with the parent as well as progeny cells. Progeny are nothing but their daughter cells, as we seen here earlier. If the parent cell has 2n number of chromosomes, the progeny will also have 2n number of chromosomes. Okay. So here the number of chromosomes is same. So that is what it is known as equational division. So that is why it is known as equational division. So in this division there are 4 stages of nuclear division: prophase, metaphase, anaphase, telophase. So here we are going to discuss in detail about prophase, what it happens in detail in prophase. So here in prophase, okay, we are going to see generally at S phase of DNA, at S phase of the cell, DNA molecules are divided. In S phase we have seen that DNA it divides from 2C to 4C. We have seen amount, for example, we have seen. So it doubles the DNA molecules in S phase, which is also known as synthesis phase, in interphase. Okay. So these DNA molecules which are doubled are not separated; they are inside a single cell itself. Okay. So initial single cell itself, they are not separated. So that is why we are going to see how it is separated and how the cell is divided. Okay. So in prophase, generally the first starting stage here, initiation of the condensation of chromosomal material happens. The chromosomal material condenses and it is visible under the microscope. Okay, the chromosomal material condenses. Okay, it starts itself to be visible in the microscope. Okay, so it starts to condense. Okay, and do you know what is condensation?
And slowly.
Yeah. Okay, so condensation like it condenses. Okay, so here at this stage, the centriole which we have seen on the human cells or animal cells, the centriole will be present in the cell division. It is also duplicated at S phase. So what does these centriole do? These move towards the opposite poles of the cell. Okay, opposite directions of the cell. If the cell is here, these move to opposite sides. Okay. So here how the chromosomes or how the chromatids are attached. Chromatids are nothing but two chromatids chromatids are attached to the centromere. Okay, a group of chromatids are known as chromosomes. So here two chromatids are attached at the centromere. We have already seen this in the cell, the unit of life, like this one. Metacentric chromosome, telocentric, that also be acrocentric, that all we studied, right? So here, this type of chromosomes which are attached to this centromere. Okay. So these are two chromatids. Okay, so two chromatids attached to the centromere. Okay, at this stage. So end of prophase is marked by, so how can you find the end of prophase? At the end of prophase, when you see a cell with a microscope, the cell do not have Golgi complexes, endoplasmic reticulum, nucleolus and the nuclear envelope. These cells do not have these organelles. Okay. So this is generally prophase. What it happens, chromosome condenses. Centrioles move to the opposite poles. Okay. And what happens, that's all. This what happens in prophase. Okay. So next we will go to the next stage, which is metaphase. Okay. So now we are going to see after prophase, we are going to see metaphase under mitotic phase. Okay. So in this metaphase, there is complete disintegration of nuclear envelope. Okay. So nuclear envelope is completely absent. When you see in microscope, it is completely absent. So that makes a starting of metaphase. Okay. So we can clearly understand that is a starting of metaphase. So since the nuclear envelope is gone, it is absent, the chromosomes spread throughout the cytoplasm. Nuclear envelope is gone. So the chromosome which is present in the nucleus spreads throughout the cytoplasm. So when these chromosomes are, since they are condensed at the starting itself prophase, so now they are clearly visible under microscope. Okay. So this phase of cell is generally morphologically studied. Okay, so morphologically how the chromosomes are present is studied at metaphase. Okay, metaphase and of M phase. So at this these chromosomes will be having two chromatids with the central chromosome, with the central centromere. Okay, so kinetochores are structures that are present at the centromere. Okay, these are structures attached to the centromere. So these the spindle fibers we have already seen in the centrioles duplicate, right? Centrioles duplicate at synthesis phase. They will go to the opposite poles on S phase we have studied. So that centrioles makes this spindle fibers. So the spindle fibers are fiber-like thing which attaches to the centriole, that is to the opposite pole, and this chromosome. Okay. So it is a fiber-like structure passing from the kinetochore to the spindle, to the opposite pole. Okay. So this these spindle fibers are attached to kinetochores. So where kinetochores are attached to centromere. Okay. So centromere is the position where it attaches to chromatids. Okay. So what these spindle fibers, after attaching to the kinetochore, they do? They bring these chromosomes at equator. Equator is at the center. Okay. So each chromatid is attached to kinetochore. There are two chromatids. Two chromatids joined at the center through centromere, and each chromatid has kinetochore. So there are two kinetochores present in a chromosome. So here each chromatid is attached to a kinetochore, connected to spindle fiber, each to the opposite poles. Okay. So this equator is also known as Metaphase Plane, because at metaphase only the chromosomes are brought to equator. Okay. So these are the chromosomes with the two chromatids and a centromere which is attached to this blue color kinetochore, and these are spindle fibers. Okay. So this is the structure at metaphase. Okay. At the end of metaphase. So we have studied prophase, anaphase, which is the continuation of prophase. Next we are going to study about anaphase. So prophase, metaphase and anaphase. This is also continuation of metaphase. After this, what happens will be studying in anaphase.